We had a little delay in some of the data that I wanted to talk about, so instead of just talking about our Poa Secunda experiments, I'm going to divide this talk up into two parts. First part, I'm going to talk about Poa Secunda, various experiments that we're doing on this species. In the second half, I'm going to discuss some of the restoration strategies that we're either evaluating, implementing, or planning to implement in the near future. So it's the middle one? This one. There it is. All right, so a little background information for all of the basis for all of the Poa Secunda studies that we're doing, which have a basic and an applied component. So kind of focusing on the applied component here. Plant local adaption is generally thought to be quite important for both plant materials development and restoration practitioners. I think this is quite important, although this is not always the case. This review article I cite here, for example, shows that local adaption is very much a species-specific and context-specific thing, depending upon where we're moving materials to and from. So restoration practitioners and plant materials developers will usually deal with plant local adaption by assigning ecozones in which plant materials, if they're inside an ecozone, they're considered to be adapted for the rest of the zone. And so ecozones, most simply, are based on climate. Some of the more sophisticated models to delineate ecozones use geographical restrictions to genetic exchange. For example, the Palouse grasslands can be quite similar to the grasslands we have here, but we happen to have the Bitterroot mountain range in between. Probably not a whole lot of genetic exchange occurring between the two systems. Some folks would call those different ecozones. And then ecozones or soils are often used as well, soil properties to delineate ecozones. And by necessity, ecozones are generally considered at very broad scales. So to develop plant materials, developers will go into different ecozones. They want to develop plant materials for that area. They'll go in, pick, get a lot of seeds for a given species, take them back, grow them, and then screen them for various traits that are deemed beneficial. So almost always it's large size tops the list or is way up there. We want to be able to harvest these seeds, so copious seed production, plants that produce very large seeds and in high abundance. It's very, very common. And then we want to be able to harvest these seeds. So most of our commercial cultivars, the seed heads won't shatter as easily as you know, if we were to go take a random pick in the population that it came from. The seed heads on our cultivars will stick to the seed heads, so they can be harvested by whatever means that species requires. And then to evaluate plant materials on different sites are almost always evaluated simply on the basis of very short-term response traits. And response traits are how a plant responds to a site. We plant it, gee, come back 2 years later, it's a nice big plant, we're good to go. And so the problem with that is that often the niche, it's very artificial conditions often that we plant restoration seeds, and those may not ever be seen again. For example, we do our best to control the weeds, and we prepare the seed beds. We really optimize conditions for these things to establish. And once the plants grow, weeds come in, they may not be able to to reproduce. And so as after the, if that's the case, then as soon as the species that we've sowed that came directly from those seeds die out, then that biotype will be removed from the system. So there's been a lot of attention lately towards analysis of effect traits rather than simply response traits. And so effect traits are not how the system influences the plant so much, but how the plant influences the system. And so if we want to develop weed-resistant communities, this really becomes important. And so we thought it is important to know how plants influence resource use, when they extract resources, and where. Like where in the soil they're acquiring nutrients, for example, and then how they influence nutrient cycles. And there's both spatial and temporal aspects here. And importantly, the species that we sow do not ever occur in isolation. They have to be able to interact with other species. Almost always in a restoration, well, in some cases in a restoration, there'll be remnant native species that the sown species will have to interact with. Almost invariably, plants are going to have to interact with weeds, and we never see it a single species on a site, so it's going to have to be able to interact in a favorable way with the species that were seeded alongside them. So in terms of plant selection, it's also getting a lot of attention that a number of studies here suggest that plants will adapt, native species will adapt to challenge from invasive species. And I've been interested in this for quite some time now. I saw Brian Mealor's, he's listed on a few of these papers at the University of Wyoming when I was there. He came out with the first paper, and I had the opportunity, pertaining to this subject, and I had the opportunity to actually crunch his data, which was just so cool. It was just unbelievable. You know, I mean, the selection processes in genotype inside weed patches versus outside weed patches. And then he and a number of people have gone on to show that those genotypes were indeed, did have enhanced capabilities to compete with and tolerate invasive challenge. So there's a number of folks that are actually actively trying to develop plant materials based upon these things that are assisted by these processes that are actually happening out in the field. So it's only been like 100 years or so since, at very most, that the native communities have been exposed to weeds, and it's interesting that in that short amount of time, we could potentially have these evolutionary events occurring to make them more resistant. So, well, the first little POA study, the first one that we started, mostly Lauren and I started this one, we sought to examine the small-scale phenotypic and genotypic variability in Poa Secunda. So there's been a little bit of work done on Poa Secunda genetic variability, but the scales are like from Washington to Colorado, and we know almost nothing about how it varies across the landscape. So we wanted to try to find what factors are responsible for this phenotypic and genotypic variation. And so we examined plant community composition, landscape position, and also soil properties. So I want to discuss a little bit about why Poa Secunda is an awesome model species to use. First of all, it's extremely ecologically important, both here, all through the shrub steppe, basically all through the west. It has a strong reason I'm interested in it and others are interested in it is that it has niche overlap is very much the same niches as cheatgrass and other annual invasive species. And so it greens up in the fall, can be active in the winter months whenever temperature allows, and is just about the first thing to green up in the spring every year. Very similar to cheatgrass. And it also senesces out almost exactly the same time as cheatgrass. So the spatial niche, it has a very shallow root system, and you think about a annual species, well, what niche space does the annual species initially fill? Well, it has to be the very top soil surface. So direct competition there as well. And Poa is kind of a little teeny guy, and it fills the interspaces in between larger grasses and forbs that would otherwise be colonized by invasives. So here's a photo of whoops, what did I do? Which one is the? Oh, that's what I thought I pressed. Okay, so this is Poa in the middle, filling an interspace. You can see there's large bunch grasses around. There's almost nothing in this interspace except little moss, a few little very small forbs. And this guy is on his way to fame in the greenhouse. Poa Secunda is also quite interesting from a genetic standpoint. It's facultatively apomictic, which means that it produces most of the time asexually. And so the offspring will, in most cases, are going to be genetically identical to the parent. But that's not always the case. It's known to occasionally outcross, and once in a while self-pollinating, and then probably even more occasionally outcrossing occurs in the species. And this can result in some of the huge differences. Mel knows all about these. She tried to key out an ampla on the ranch, which is also a Poa Secunda, except it happens to be about that big. And she had fits over that one, keying out something that's supposed to be that big. That's due to rare outcrossing events that result in huge differences in morphology. It's polyploid. This probably allows it to be more plastic out in the world, but unfortunately this does complicate genetic analysis. Really wanted to do microsatellite analysis to get at these outcrossing rates, and that would be quite complicated to do. I'm not sure it could be done. Has a strong potential for failure, so we kind of didn't go there. So what we did, well, what Lauren mostly did, is go to 14 different locations across the ranch and actually physically dig up 12 adult plants from each location. So we stratified the sampling design, first of all, just to take up as much space on the ranch as we possibly could. We were interested in examining variability at the scale of the ranch, and so we went far north and far south, far east, and far west. We also were interested in determining the influence on plant communities, so the attempt was made to sample within different plant communities, and also we tried to sample an elevation gradient. She has perfect teeth. Perfect teeth. I had to include this. But we had to sample in the fall when Poa was not active, and so everybody doing that was donned in the usual fall fashion at MPG, which is blaze orange. So we brought these back to the greenhouse, split each plant in two, and potted them separately in different plots. I keep saying we, actually, it was once again Lauren heading all of this up, but then after a few weeks growth, leaf numbers on each clone were counted, and then one clone from each pair was challenged with cheatgrass, and this is simply just sprinkling cheatgrass seeds around each clone. And then these plants were grown for 3 months, and then we used the difference between of leaf numbers between the unchallenged and challenged treatments as a proxy for cheatgrass tolerance, Poa Secunda tolerance to cheatgrass rather. And then for competitive ability, we simply cut cheatgrass biomass at the soil surface, dried, and then weighed them. So high competitive ability, low equates with low cheatgrass biomass per pot. So here's some results for tolerance. You can see we got a fairly significant relationship. There's definitely some variability between populations. Competitive ability is quite pronounced, though. We have over a threefold difference in cheatgrass biomass between pots. So, gosh, these guys appear to, vary quite a bit in their ability to compete with cheatgrass. So, what factors are responsible or could be driving these phenotypic changes? We looked at plant community composition. Lauren once again went out and analyzed vegetation on each site. And this is a very interesting figure and also very, very unsatisfying. And unsatisfying from the point of view that almost all of these had very similar non-native species cover. However, the ones that had the greatest or the least competitive or challenged by non-native species had the greatest cheatgrass biomass, therefore the lowest competitive ability, and the same on the other end, the inverse on the other end. So there is seem to be a trend there. We're going to nail this, I think, though, with we're doing another POA study very similar to this with the EMP sites, and on those sites we have 19 different sites, and we have paired native and invaded communities right next to each other. And we've collected POA from all of those, and so they'll be much, much cleaner than this. We should be able to we will have highly challenged and unchallenged sites. So that should be interesting. Okay, spatial proximity is probably going to be important, or would be important. One would think it would be anyway. And so just did distance matrices of tolerance and competitive ability and compared them with physical distance matrices using Mantel tests. And sure enough, tolerance was significant. So the closer populations were collected to one another, the more similar they are in tolerance to cheatgrass. Competitive ability, almost significant. It's one of those cases, well, gosh, if we had one more rep or something, it would go over the line. But there's a pretty strong trend there. So that's kind of cool. And then, for landscape features, we regressed elevation versus competitive ability, and there's a little leverage going on here on these sites. But there's pretty nice a nice trend. So as you go up in elevation, the tolerant or the competitive ability, surprisingly, I would have thought it would be the other way around, would increases. And so low elevation sites, historically the most disturbed, I don't know. It's a little bit tough. When we're assuming, although, that the only thing selecting for these genotypes is the invasive species, or these invasive species, cheatgrass, it could potentially be other invasive species, who knows. We should be able to tease this out over time, I hope, anyway. We're going to have over double the data for sites within a year, and so hopefully hopefully this will become clearer. And then I made a solar radiation map of the ranch. So solar radiation, it is interesting in that it includes both slope aspect, but then also nearby topographical features that could block sunlight. And so I think it's much more informative than just using slope and aspect. So made this model, and then for each month of the year, calculated solar radiation, and then compared that with the tolerance and competitivity on each site. So there wasn't a significant relationship with competitive ability, but tolerance was fairly strong. I mean, I was a little bit surprised. Strongest relationship was for March. I don't remember exactly what that p-value was, but so solar radiation low and across the winter, fall, and spring months resulted in, well, correlated with increased tolerance. So cooler sites, shorter growing season, milder conditions seem to equate with tolerance. And then also the plant communities change quite a bit across different aspects or with solar radiation. So that might come into play as well. And here's where I was going to discuss the genetic analysis, but this is the data that we do not have. Alana Shaw, back here, she has been quite a trooper on this, cranking away trying to get this data out, and it's certainly not her fault that we don't have this data. The Murdoch Center has delayed us, probably over a month, but we actually did get like about half this data 2 days ago, and didn't really allow us too much time for analysis. So anyway. And then also I wanted to talk about seedling, or Poa seedling germination traits. I planned to do this. It has kind of been stalled. So what do we want? We only looked at adult plants in the study. I've only discussed adult plants so far, but we really want to be able to compete with invasive species, annuals anyway. We want our seeds to be able to germinate at a very low temperature, just like cheatgrass does, so it can get out there and start competing for things. And then in terms of root morphology, we want these seeds to grow fast after they germinate. That's what cheatgrass does. It grows fast. It can go find nutrient or resource pockets before the natives do, and monopolize these resources. So that is in the works. So we planted these, we've moved all these populations out into the field, and so they're all growing under the same exact environment. We collected seeds the first year, we collected seeds from these last year. We're going to collect seeds again this year, and then compare to see if these actual functional traits change over time depending upon the maternal environment. There's getting to be a lot of evidence for this sort of thing occurring in the literature now, so looking forward to that. Okay, the third, or what is it, fourth POA study, I'm just going to mention. This is being done by Molly Hergut, and who's a graduate student of the University of Wyoming, and she works with Christina Hubbard, which is a she calls herself a restoration geneticist at University of Wyoming. And so what Molly's doing is she has taken 3 of our populations, the one with the lowest, medium, and highest competitive ability, and then 3 commercial cultivars that would be most likely species that we would use for restoration on the ranch. And in fact, we are using these biotypes for restoration on the ranch. She took all of these, planted them. Each one of these little grids contains a few POA seeds, and then half of them were challenged with cheatgrass and half of them were remained unchallenged. This is actually a huge little experiment. It's going to require a huge amount of work this spring, and so we're hoping to get Molly back here pretty darn soon. So, and I hope she's going to step up to the plate on this one. But okay, so that's the. How much time do I have, Philip? Okay, 20 minutes. All right. So yeah, this is where I switched track, so weed control and restoration is probably the biggest consideration. I certainly think it is. So we're this year mostly interested in converting the crested wheatgrass fields to native communities, and so we think for this purpose and also for annual weeds that pre-emergent herbicides are probably the tool of choice for this purpose. But unfortunately, most pre-emergent herbicides not only will inhibit seedling recruitment from the invasives, but also most of the things that we plant in with them. And so if we could come up with a way to protect just the native seedlings from the herbicides, then they would be able to fill in the niche space. I should have mentioned that most pre-emergent herbicides, they recommend a plant back time, and this varies from herbicide to herbicide and the amount you use. But some, you shouldn't be planting back for 6 months until the herbicide wears off, or a year, or even 2 years. And then you can go in and plant. But the trouble is that any weeds that are remaining there are also freed from control, and they can compete directly with whatever species you're sowing. So if we can protect the seedlings from herbicide, they can grow free without any kind of competition or very minimal competition. We could probably get some fairly substantial growth first year. The niche space would be filled in, leaving a lot less for weeds in the future. So one thing we're trying, here's a posed picture of Lauren spraying activated carbon right down seed furrows. So activated carbon is highly absorbent. It absorbs a masopic like crazy, and that's what we're using here. So we took a diverse native seed mixture and drilled them across the hill above the orchard house, sprayed activated carbon in 3 different amounts down these furrows, and then came along and sprayed 12 ounces per acre of imazapic, which is the highest amount on the label over the top of this. And we'll go back and evaluate in the spring to see what emerges and what is controlled. So here's a greenhouse experiment. We just completed a little test in the greenhouse. So this is slender wheatgrass. On the right is 0 AC treatment, and then going from right to left, the amount of activated carbon increases per pot by 0.5 grams. And so you can see that there's no growth in the no AC treatment. We're getting some minor growth. These are quite sick little plants though in the 0.5 gram per pot treatment. These probably would not make it in the field when exposed to drought or drying or any other severe environmental conditions. But everything from 1 gram to 2.5 grams seem to be completely protected from the herbicide. So I had heard of this. There's old, I get this idea from this very old paper that mentioned using AC in agriculture. And this was very old, and I didn't see very much. And then I heard about these guys in Oregon that are planting native seed production fields, and they're using this technique. So you do not want weeds in your native seed production fields. really decreases the value of the seed. But then I couldn't find who these people were, and I told this guy that was using this in agriculture, but he knew some connections, and so I got hooked up with a couple of guys that are doing this in Oregon, and then they subsequently gave me some guys that are actually using this in restoration in Colorado and in California. So it is being used, and most people that are using the technique are quite excited about it. So he gave me some kind of schematics for this is a seed drill, and so what he's got are these little, he's spraying an activated carbon slurry right down over the top of the furrows. So as you go along, this would not work for our purposes though, for a couple of different reasons. They're able to maintain a constant temperature over this flat field. We're going over hill and dale. We're going to have to adjust the flow rate on this, so we're probably going to have to use radar or a GPS-based flow control. And then also, he's using these 2-inch wide strips of AC, and well, we have to turn. And then he sent me this picture, and you can see, yeah, he's going around like a center pivot or something in circles, so that would complicate things. We should be able to go in a single direction or in straight lines. So I'm not, there's some problems we need to address before we propose to do large areas with this technique, and there's still a lot of work to be done to see if it's going to meet all expectations. But certain things that I can see it could be a real big problem are any kind of service litter. Well, we did that little thing that where Lauren was showing spraying the AC in that field. There was a lot of service litter there, and that could be a really bad thing. We need to see how much of that we need to remove, if not all of it, to get good herbicide contact. The other thing is that any weed seeds underneath the AC will be protected, as well as the native seeds. And okay, so how much crust can we live with? You know, and so we want to do some experiments right early in the spring. We're not going to kill all the crusted, but I think, you know, I think we have a good chance of knocking out like 95% of it or so. But even 5% of the seed bank, which could be quite extensive, might be enough to complicate things. And I wasn't sure I wanted to mention this or not, but these are pretty cool. These are, Bo mentioned these things yesterday. These are the herbicide protection pods. This is, well, Matt Madsen, he came up with these things. They're little agglomerated seeds in a little block of AC, among other things. And so the idea is that we can go out and broadcast seed, and then they'll be protected from pre-emergent herbicides and grow nicely. So we did a greenhouse study in tandem with Matt here. You can see the little black blobs on the top of some of these. So we put these herbicide protection pods out and then sprayed with various amounts of masopic, and then also just coated seeds very lightly with AC, and then had naked seeds as a control. So here's some of the data from that. This is cheatgrass on this side. If you can't read that, plant height, plant biomass, and well, gee, you know, our lowest amount of imazapic basically cleans cheatgrass's clock, just complete wiped out. But in contrast, on this side, if the seeds are unprotected, there's 100% mortality almost. But if they're lightly coated, we get some protection from the low rates of masopic, and some from going on up. The blue bunch doesn't fare quite as well. But on the other hand, on the herbicide protection pods, this even suggests that they do better when seeded with the pod than they do just seeded naked without any chemicals added. That's not significant, but it's kind of interesting that they all have a higher value than the controls. The reason I'm not potentially I'm really excited about this, but what we need is enough of these to cover huge areas. I mean, we could do little, I don't think that's going to happen for a while. I mean, we can run out and do little test plots and everything else, that's great. But when we want to restore large areas of the ranch, these things are going to be limiting for a while. So here they are. Oh, the other really cool thing about these is that they also address other potential seeding problems that we had. Lauren, once again, went out and not with these particular pellets that have AC in them, but pellets that have exactly the same matrix except for the AC, and evaluated seed predation rates. And the seed predators on the ranch do not touch these. This is very important since broadcast seeding we can lose 90 or better % of our seeds in some cases to seed predation. And these are hydrophilic, so that's also, how much time do I have now? 5 minutes. All right, and have to move right along then. So as I mentioned, we've got lofty goals this year, and that's elimination of basically most of the introduced grass lands and the category 4 areas. By category 4, I mean that areas that have almost no native vegetation that we deem is worth salvaging. So we can get pretty heavy-handed with these these areas. So yeah, we've proposed these areas in black to do that AC thing. As I said, we're not sure. We need to continue to evaluate it throughout this summer. Depending on how well it works, we may not do these areas or expand to some of these other areas. We have got 2 or 3 different strategies depicted here. Different bridge species, for example, and including chemical fallow. And then I've made a new team. I saw a team in the teams folder. We had POA, and yeah, we should be Team POA. And so, yeah, there's quite a few people actually working on this, and a lot of people that don't actually even know that they're involved with this. So Lauren, of course, Alana, I've already mentioned Molly and Christina, and this is part of this is part of the EMP project. So Elva and Philip are both EMP teamers, and so yeah, they've got there's a lot of overlap in the data will be used, et cetera, for that. Vegetation crew, you guys probably didn't have a clue, did you? But yeah, we're hoping to do geography for POA of the ranch, and so we'll be using a lot of you guys' data to, yeah, in that regard. And of course, Jeff Clark and friends, and then there's been other people that Lauren's been able to con into helping with the different aspects. We saw Heather for one up there, and yeah, I guess that's it.